Ever think about how a tiny machine, like a single bacterium, makes life or death decisions every single second? It's pretty amazing. Today, we're diving into one of the coolest examples of this, the Lac Operon. It's this incredibly smart genetic switch, and honestly, it has a lot to teach us about survival and just being efficient. I mean, seriously, would you? Of course not. That would be a crazy waste of time, money, energy, you name it. Well, believe it or not, bacteria face the exact same problem. Making the tools, in their case enzymes, to digest food costs a ton of energy. So it begs the question, why on earth would a bacterium bother making enzymes for a sugar that isn't even around? So let's talk about our little bacterium, E. coli. One of its potential meals is lactose, the sugar in milk. But to use it, it has to build a whole set of special enzymes. And here's the catch. Making proteins is one of the most energy-draining things a cell can possibly do. So the bacterium has this massive problem to solve. How does it make these enzymes only when lactose is actually on the menu, and not a single second before? And it turns out, bacteria are absolute pros at managing their energy. They basically have two kinds of genes. You've got your constitutive genes. Think of these as the ones that are always on. They're for basic housekeeping, like keeping the lights on. Then, and this is the cool part, you have inducible genes. These guys are off by default. They only get switched on when they're really, truly needed. And that is the energy-saving trick that makes this whole lactose system so brilliant. Okay, so how does a bacterium actually build this smart switch? Well, it uses this incredible piece of natural engineering called the Lac Operon. It was discovered by François Jacob and Jacques Monod, and what it is a set of genes that are all controlled as one single unit. You can think of it like an automated control panel for the bacterium's entire kitchen, making absolutely sure nothing goes to waste. All right, let's look at the parts of this control panel. First up, you've got the structural genes. These are basically the recipes for the tools the kitchen needs. There are three main ones here. LacZ, that's the recipe for the enzyme that actually breaks the lactose down. Then there's LacY, which makes an enzyme that acts like a doorman, bringing the lactose into the cell. And finally, LacA, which has a role that's a bit more of a mystery, but we know it's there helping out. Now, every good cookbook needs a start here page, right? For the Lac Operon, that's the promoter. It's a specific bit of DNA that's basically a giant start button. It's the landing pad for a molecule called RNA polymerase. That's the molecular machine that's going to read the genes and get the whole process started. Okay, so we have a start button. But what if we want to prevent someone from just pushing it whenever they want? That's where the operator comes in. If the promoter is the start button, think of the operator as a security lock placed right over it. It's a tiny stretch of DNA that can physically get in the way and block the machinery. This little lock is really the heart of the whole on-off switch. So who has the key to this lock? Or rather, who is the lock? Well, that would be our security guard, which is actually a thing called the repressor protein. This protein is made by a totally different gene, the regulator gene, and its one and only job is to find that operator lock and clamp down on it, keeping the whole system shut down by default. All right, so let's do a quick recap of our smart kitchen, right? We have the recipes themselves, the LacZ, Y, and A genes. We've got the chef ready to cook, that's our RNA polymerase, and we have the whole control panel, a start button, the promoter, a lock on that button, the operator, and a dedicated security guard, the repressor, whose default job is to make sure that lock stays firmly in place. Okay, all the players are on the field, so let's see this thing in action. First, let's look at the default state. What happens when there's absolutely zero lactose around for the bacterium to eat? It is so elegant and simple, you're going to love this. The repressor protein, our security guard, is on duty. It binds super tightly to the operator, the lock. And by doing that, it physically blocks the RNA polymerase from even getting to the genes. It's like a roadblock. So long story short, transcription is off. The kitchen is closed. No energy is wasted. It's a perfect example of what's called negative control. But what happens when things change? What happens when a big, juicy molecule of lactose finally shows up on the scene? This is where the magic happens. When lactose gets into the cell, a little bit of it gets converted into something called allolactose. And this allolactose acts like a special key. It finds our security guard, the repressor protein, and binds to it. This binding changes the repressor's shape, kind of like giving it a hug, and makes it let go of the operator lock. The security guard is officially distracted. With the guard gone, the path is wide open. RNA polymerase jumps on the promoter and starts transcribing the genes. The kitchen is now open for business, pumping out all the enzymes needed to chow down on that lactose. Okay, I know what you're thinking. It's the classic chicken and egg problem, right? If the system is turned off, how does that first molecule of lactose even get inside the cell to start the whole process? Great question. The answer is that the system isn't 100% airtight. It's what scientists call leaky. There's always a super low level of background production going on. So there are always just a few of those dormant enzyme molecules hanging around, ready and waiting to let the very first bit of lactose in. And just when you think this whole system can't possibly get any smarter, there's another layer, another twist. See, this bacterium isn't just efficient, it's also, well, it's kind of a picky eater. It has a favorite food, and it will always go for its favorite if it's on the menu. And that favorite food is glucose. It's a much simpler sugar, way easier for the cell to use for energy. So even if there's plenty of lactose around, the cell asks another question. Hey, is there any glucose here? If the answer is yes, the cell will chow down on the glucose first and keep the Lac Operon turned way down. This whole idea is called Catabolite repression. It's basically the cell saying, why would I bother cooking a fancy 3-course meal when there's perfectly good fast food right in front of me? This table really lays out the logic of our picky eater perfectly. Let's just walk through it. Okay, top row. You've got glucose, but no lactose. The repressor lock is on. Simple. The system is totally off. Makes perfect sense. Second row. Now things are interesting. You've got glucose and lactose. The lactose takes care of the repressor, so the lock is off, but because the cell's favorite food, glucose, is there, a special accelerator protein doesn't bind. So the system is on, but it's kind of sluggish, just a very low level of activity. Third row. No food at all. No glucose, no lactose. Well, the repressor lock is still on, so it doesn't matter what else is happening. The system is off. And now, the last row, this is the sweet spot. No glucose, but plenty of lactose. The repressor lock gets removed, and that special accelerator protein binds, basically slamming the gas pedal. The system goes into high gear. See? The kitchen only ramps up to full production when its favorite food is gone and the backup option is the only thing on the menu. So there you have it, the Lac Operon, a truly fascinating little system inside a tiny bacterium. But why do we care so much about it? Why does it matter? Let's zoom out for a second and look at the big picture. Well, the discovery of the operon was a genuine game changer in biology. It was the first time scientists had a clear, mechanical example of how genes could be switched on and off. It proved that genes aren't just these static, dusty blueprints on a shelf. No, they're dynamic, responsive switches that react to the world around them. And that concept, regulated gene expression, is everything. It's universal. I mean, think about it. Every single cell in your body, from a nerve cell to a skin cell, has the exact same DNA blueprint. So why are they so different? How can one be a muscle cell and another a liver cell? It's all because of gene regulation. Different switches are flipped on or off in different cells, and that's what gives them their job. The very same logic that was discovered in this little bacterial kitchen is the same logic that builds and runs, well, you. So I'll leave you with this thought. If a single humble bacterium has such a sophisticated and elegant genetic control panel just to manage 3 genes, just try to imagine the kind of programming, the sheer complexity that's running the show for the more than 20,000 genes inside of you right now.